Hello, everyone. Thank you for joining the monthly national NBS Pilot webinar for the July monthly call. I'm Kevin Wilhelm, and I will be hosting this webinar. Just a reminder to please mute yourself as you join. I believe I muted everyone initially, but just keep an eye on that. And here are the dial-in numbers as well. And today we will be getting back to the normal agenda after the presentation by Dr. Piero and Piero Ronaldo and their group at the Mayo Clinic last month, which is also posted on our YouTube page if you would like to go back and review that. And so today, I would just like to start with any introductions from any state labs out there that are new to the call or would like to introduce any new members that are also a part of their team. Nope. Okay. Okay, so I guess we'll just get started with the pilot sites. Is there anyone from the New York team that is on the call and would like to update us on their progress?
Hi, this is Michelle. I have, I don't think Joe is on, but I think Colleen is on. I have some numbers here. Let's see, must be from the last time. I don't know if there's any change, but I will go through the numbers I have and see how they compare. For Pompeii, we've had 4 additional referrals. So we have a total of 164. The number is still somewhere around 1.4 million screened. We have 9 infantile, so I guess that one of them was with that. We have the 1 late onset, but no cardiac involvement, and I think that's an old one. 37 are probable LOPDs, so those are ones that have 2 pathogenic or likely pathogenic variants. None of those have become symptomatic as far as we know. We have 35 other possible LOPDs. So as you can see, our infantile rate is quite low. And that category is 1 pathogenic and likely pathogenic variant, and then a variant of uncertain significance. So they get parsed out as possibles. We have 13 that are uncertain, and then 69 carriers.
69 carriers?
Yeah.
And how many screened as well?
The number I have is at 1.4 million, so it's still in that same kind of range, I guess.
Okay, great. Thank you.
For NPS 1, I have 410,000 screens so far. We've still had 20 referred. 3 of them had 1 pathogenic or likely pathogenic variant plus a VUS. As far as I know, we haven't had a classic yet. Out of those 3, 1 had elevated GAGs, 1 was minimally elevated, and then 1 was normal for GAGs. So out of those 3, 17 babies were carriers, and then 47 were not referred. They were positive on the biochemical, but only had pseudodeficiency, so they're just reported.
Okay.
Moving along, ALD.
And what was the 17 there, sorry?
17 were carriers.
Carriers.
Yep, and
okay.
47 were pseudodeficiencies, so no referral. Yep.
Okay.
ALD, we have about 1.62 million screened. We've had 116 referrals, and of those, 59 are boys and 57 are girls. It's still running about 50-50 split for female carriers versus boys that actually carry a diagnosis.
Okay.
Okay. And lastly, SMA. We've screened about 410,000. We have 18 babies referred. Out of those, 9 of them have 2 copies of SMN2. 7 have 3 copies. And 2 have 4+ copies. And almost all of them that are in the category to get treated are on the gene therapy. And that's it.
Okay, great. Thank you for that.
You're welcome.
Does anyone have any questions for Michelle?
Hi, this is Bob Vogt at CDC. And Michelle, I did have one question about the XALD. That, as I recall, is a screen looking for metabolites. And wondering, are the carriers picked up with the same? Do you get 100% of carriers in your female population?
We don't think so. It is running even between girls and boys, but girls have 2 X's, so we expect more girls. We wouldn't know, we don't know who we missed, because if they don't have the biochemical screen positive, then we wouldn't, we wouldn't know that they were a carrier we missed, because we only sequence the kids after they're positive for the biochemistry.
Sure. And is the profile such that it looks like a carrier state from the metabolic ratios and all? Can you see that in the chemistry?
Some of the girls have fairly high C26 values, I think. Joe's more the expert than I am on looking on that sort of a day-to-day basis, but some of our carrier girls have relatively high C26s, I think. It's not like, half of the boys or whatever.
Okay, thank you. Thanks very much.
Mm-hmm.
Yeah, this is Stan from California. We're seeing the same thing. The girls have high C26 in California.
And then, they're probably the ones we're picking up because of that, right?
Yeah, and they are carriers. You'd expect a 50-50 ratio. After we changed our cutoff and raised it, we're seeing more girls than boys, which is interesting.
Hmm. Is there any phenotype, any expression of the carrier state in the girls? Other than metabolic?
Later on.
Years later, supposedly, yes.
Okay.
Right.
All right. Thank you all.
Yeah.
Mm-hmm.
Hi, this is Mary Schroth with Cure SMA. I just wanted to verify the number of screened for SMA. It's 110,000 or 410,000?
Is it wrong on there? It's 410, yes. Sorry. Good catch.
410. Okay, good. Thank you.
Yep. Mm-hmm.
Okay, great. Thank you. Okay, is there anyone from Georgia on the line? Anyone from Georgia?
Well, in a technical sense, I'm from Georgia, but I'm not part of the Georgia newborn screening. Could not speak on their behalf.
You can tell us what you've been up to at CDC, Bob.
Yeah. Yeah, I went to high school here, so I can speak about some things for Georgia, but not the newborn screening program.
Okay. Well, hopefully we can catch up with them on the next call. Is there anyone from the North Carolina team on the line that would like to update us?
Hey, Kevin, this is Blake. Can you hear me?
Hi. Yep, I can hear you.
We have screen 9,111. We're inching closer to our first big milestone, 10,000 screens. Still no positives. We will be continuing through December. And we're continuing to push recruitment right now. We have, hopefully, with some hospitals opening up some additional alternative recruitment methods that we're going to start using, but we're still very focused on increasing our uptake rate here in the state. And we're also working on adding a new condition, Duchenne muscular dystrophy, to the panel right now. We're going through the verification process with the instrument and preparing to start screening for that, hopefully by the end of the summer.
And Blake, this is Cindy, Cindy Powell from UNC. I just wanted to make sure. So the total screen number is 9,111, correct?
Correct, yeah. Sorry,
Okay.
I didn't see that mistake.
Sorry, I'm just getting all my numbers confused today.
No problem.
Okay, and what instrument are you using as well for the DMD pilot?
The GSP by PerkinElmer.
GSP. Okay, great, thank you.
Mhm.
Okay. And then I guess we can move on to discussion and report from other states. As many of you know from the last call, we had a presentation from the Mayo Clinic team regarding the Pompe and MPS1 second tier test. And next month, we will be having a presentation from Dr. Mary Schroth at Cure SMA, looking at what they're trying to do with SMA. And Mary, are you on the line? Would you like to talk a little bit more about that?
So I'm going to talk on August 6th. So I'm not sure what you would like me to say, Kevin. I'm sorry.
No
But
worries.
yes, I just wanted to have an opportunity to share with people what we've gathered from all of you, and what we're aiming for, and just the impact of newborn screening. How very important it is to have newborn screening for SMA implemented throughout the country. So I'll be sharing with you the clinical side and what happens. And if there's anything that you would like us to address, please let me know. During our time next month. My email is mary@curesma.org.
Great, thank you.
Thank you.
Yeah, and we'll go ahead and nail down a little more of what will be presented and such. And we'll post that to the APHL listserv the week before, just so you guys get to read the abstract or a quick overview of what we'll be talking about next month. Is there anyone else? Oh, sorry, go ahead.
Yeah, I'm sorry to bust in, but I did want to mention this. 2 years ago, the CDC Foundation received a donation from Biogen to distribute directly to newborn screening programs as supplemental funding that was highly flexible as far as utilization. And that money was distributed, and we are now in the second round of that distribution process. There's a total of about $600 and some thousand that can be distributed with individual grants ranging between 50 and 200,000, I think. The announcement is on the CDC Foundation website, and I should have sent that to you all before this, and I'm sorry I didn't, but I'll send it during the call here. So if we do want the programs to know about this, it's an easy application process, and the money is highly flexible as far as utilization. And I think it was helpful for the first round of programs that received funding. So we'd like to get it out. We'd like to, and it's a rolling application process. It is not competitive in the sense that there's not a deadline and then everything is reviewed. Each application is reviewed as it comes in, and applications are received up to the point where the funding has been exhausted. So it's kind of a first come, first serve thing. And we would very much like to see programs that are in the process of getting ready to screen. This is not meant to support the initiation of screening. It's meant to catalyze, accelerate the pace at which population-based screening can be reached. So we would very much like to hear from programs that are in the process of, and when I say we, I should say the CDC Foundation our branch and the agency, CDC, is actually not involved at all in this. This money came directly from Biogen and is being sent out directly by the CDC Foundation.
This is Mary Schroth. Just to clarify, just to clarify, is this specific to SMA or is this more global for newborn screening?
This is Mary. Hi, thank you for asking. Yeah, it's entirely specific to SMA.
Okay, thank you.
Great. And Bob, if anyone's interested, where should they reach out or look on the website?
They can certainly email me, and I am teleworking, as all of us at CDC. We're still in phase 1. So I have learned how to telework, first experience in my life of that sort of thing. Certainly you can email me. It's rvogt, R-V-O-G-T, @cdc.gov. And as we're moving through here, Kevin, I think I have Amy's email address extremely handy. So I'll send the CDC Foundation link to Amy, and then it could be distributed to everyone. Is that okay?
Yeah, thanks, Bob.
Okay. Well, thank you all. We're hopeful that this will accelerate the process. You know, I always think about with the birth prevalence and so on, my estimate was that every day in this country, there is a newborn born with SMA approximately. And so it really, the time frame on this, and because of the severity of the disorder and all, is an important feature of the public health effort.
And Bob, I would just add to that the results of the 2 treatments that are FDA approved to intervene pre-symptomatically have made such a dramatic difference in outcomes that I'm very passionate about this, that identifying these babies early and doing these and providing this treatment just has been groundbreaking, life-saving. We'll talk more about it next month. Thank you.
Thank you, Mary.
Great, thank you for touching on that. Is there anyone else, another lab out there that would like to update us on their progress for SMA or any other pilots that are going on?
Hi, this is Bonnie Chad from Florida. Can you hear me?
Yes.
Yay, I finally got my settings right. I've been struggling with them. Yes, we went live with Pompeii and NPS1 mid-February, and we have mandates in Florida that we have to meet as far as when they go live. We had a mandate to go live with SMA by August, but the legislature moved it up to May. So we finished going live 15th of February with LSDs, and we went live with SMA on 4/27. And since then, we have 4 positives within that time frame, which was surprising to us, but and also one that was sort of retroactive. The baby was born in April, but was showing signs, and so it was within a few weeks of birth, they sent it in, and so we've identified 5 so far. 3 are still waiting for SMN2 copy number. The other 2 are 2 copies. So, and they're in the line for treatment now. So it's going extremely well for how rapidly we've been onboarded here. So I don't have precise numbers for other disorders, but that's our most recent findings. Our population is in volume is similar to New York, so it'll be interesting to see how that goes as we continue through the year.
That's great. Thank you for the update. Sounds like everything's going well down there.
Well, it's going fast, but yeah, it's going pretty well. Thank you.
May I ask how many have you screened?
We have a throughput of about close to 5,000 a week. So I'd say we've screened in the vicinity of about 40 to 50,000 since we started. I'll have better numbers when I get back. I just haven't prepared it that way.
Okay. Great, thank you.
Hi,
Is there anyone else? Oh, sorry, go ahead.
This is Kim Piper from Iowa. We started our SMA implementation pilot yesterday and things went smoothly. During our test validation process, we did identify, we had a positive result for one of our control cases. And that was on a 30-day-old newborn. And we sent the test to Minnesota. Since we weren't validated yet, we sent the test to Minnesota. Actually, Dr. Travis Henley drove it up there to Minnesota, and they were very helpful, and they tested it overnight and it returned a positive result again. Another positive thing that came of this is that this was a baby that was identified prenatally. So the program was already aware of this baby, and the baby was receiving treatment due to an identified sibling.
Wow, that is a story for the books.
It is, it is. And there's even more to that, but I can't get into it. So.
Wow.
Yeah.
Lucky.
So just
great for the family.
Yeah, it was great for the family, and it was great. I mean, it showed that our test worked. So, I mean, we had it found that baby. So
Great. And that test was confirmed by Minnesota, correct?
Right. Yep.
Okay. Great.
Many thanks to them.
Congratulations and welcome to the world of SMA newborn screening.
Yeah, thank you.
This is Mary Schroeder.
Just to note about treatment, the sibling of that baby got the gene therapy, and then the newborn is getting nusinersen.
That's interesting. You probably can't say much more about the particulars of that, but did anything? Are you aware of any particular reason why the choice of therapies was different in the two children?
Yeah, we do. And maybe Dr. Schroth will speak to this next month too, but apparently there is some, oh, now I have to make sure I'm saying this correctly. Anyone that is around a child who's been getting the gene therapy treatment can develop antibodies towards that, or not really antibodies, but can develop.
You're correct.
Okay.
So for some infants, and it's a very, and I will touch on this next month, thank you. A very small percentage have positive antibodies to the AAV9, which is the vector that the gene therapy is delivered in. And if they have elevated antibodies, the therapy is ineffective. Most of the time, those antibodies actually due to maternal antibodies, and they get repeated testing. So it's possible that the baby may receive gene therapy in the future, but they can't until their antibody levels are very, very low.
Yep, and since this baby was identified prenatally, that was the route that they chose to go, which I think is very interesting.
That is extremely interesting, and thank you, Mary, for clarifying that. I never thought about that. I haven't heard it come up before, but is it possible that the gene therapy in the first baby was responsible for the antibody response in the mother? Is that?
No, that's highly unlikely. The antibodies are shed at a very, very low level during the first 30 days following treatment, and then they're essentially gone. And parents are advised to be careful in how they get rid of the dirty diapers, for example. So most of the time, it's maternal antibodies, and adenoviruses. It's in the community. People are exposed. The older you are, the greater the likelihood that you've been exposed or had it or have antibodies. So typically the intervention is that mother stops breastfeeding for 30 days or more, and the antibodies are rechecked.
Thank you, Mary. That's invaluable information. My recollection is that when the Ohio State group did a pilot study, I think they removed almost a third potentially eligible childbearing women because of the presence of antibodies. So it's not a trivial number, is my understanding. Is that your impression?
So for the Ohio State single site study, they had no babies, and I can only speak to the babies. No babies that were excluded because of the antibodies being elevated. However, in the multi-center trial that followed up, I believe that's the STRIVE trial, there were 3 babies that were excluded because of antibodies, AAV antibody levels at the time of screening.
Thank you. Thank you all very much.
This baby's sibling was also about a year old.
Okay.
And this pregnancy was unplanned.
Thank you.
Part of the joy of newborn screening.
But they knew in advance. I mean, but what you shared with this group is that they had prenatal testing and they were aware of the baby's diagnosis, and it was for the newborn screening, it was a validation process.
Yes, definitely. And both of those children have responded remarkably well to their treatments.
Good. I mean, the clinical data about pre-symptomatic treatment with both the gene therapy and separately a separate trial looking only at the nusinersen, both have had extremely favorable outcomes, which again is why we as a patient advocacy organization are just pushing so hard to get newborn screening throughout the country. And again, thank you for letting me share with you. I so appreciate it, and letting me on these calls.
You know, it would be nice to write that up as sort of anecdotal report, I guess. But I don't know. I'm sure that's happened with other conditions and all, but I hadn't heard of it happening with SMA so far, where a prenatal test had already been done, and then it came into the newborn screening domain. So that might be the first, and I don't know how often it will happen in the future, but it just shows how common this is, I think, to some extent. But if you all could write that up, maybe for if there's a state medical journal or one of the larger journals, or a letter to the editor or something, I think that's very interesting.
Yeah, we definitely will consider that.
Terrific.
And this family has been very supportive and whatever you guys need us to do for SMA screening, we'll do it. So they're good advocates.
Okay, great. Thank you all for sharing that. Is there anyone else that would like to come in and update us?
Yeah, hey, can you hear me?
Yep.
Yeah, hi, my name is Vineet from Illinois, newborn screening. We actually just went live with our SMA on Monday, so yay. And so everything's been going pretty well as far as screening this week. We actually, throughout our validation back in February, we actually did encounter a baby with SMA who screened positive in our hands. We sent the specimen to Wisconsin for confirmatory testing, and they also confirmed it as well. So it was kind of fortunate that when we were doing our validation, which consisted of about assaying of approximately 10,000, we just so happened to pick the 3-week window of our newborn screening intake that this baby was selected in. So it was actually pretty fortunate that we were able to screen, encounter this baby during our validation of the assay. And we're also, we're multiplexing the SMA with our SCID assay, so I think a lot of people across the nation are doing the same thing as well. So yeah, just kind of wanted to give an update on that. As far as that baby, I know diagnostically determined to have the 2 copies of SMN2. And I think there was, in talking to some of the clinicians, I do know that they had encountered some difficulty with the insurance companies going back and forth. So we're actually going to be also going forward to investigate doing SMN2 determination as well. Right now we just do the SMN1 screening. So just kind of, we might do that with Illinois kind of down the road as far as maybe doing that as well to help the clinicians as far as building a better case with the insurance companies to help get the funding for the treatments going forward with any positive babies.
Great. And so was that insurance difficulty around getting them to pay for the SMN2 testing or through treatment?
Through treatment, actually, yeah. I mean, it was like kind of like, I was talking to the clinician in charge. I mean, he was just kind of, he was just frustrated with it, how he had to go through, several rounds of peer-to-peer review with the insurance companies. But ultimately, it passed. But it was just, he had made the argument that if we're able to get SMN2 screening, information alongside any positive SMN1 screening, it would help their case tremendously. So we're definitely going to look into that as well at some point down the road.
This is Bob. Congratulations to you all. It's wonderful to hear the program has accomplished this. May I ask what method you're using? I'm wondering if it's the Perkin-Elmer commercial method.
No, this is I think the CDC one. We got the design from, I guess, from CDC, from what I understand. So we're just multiplexing that in with our current SCID assay, which is basically an RT-PCR.
That makes me even happier to hear. That's terrific.
Thank you and congratulations, Illinois. We're so happy to have you on board with newborn screening. This is Mary Schroth with QRSMA.
Yeah, thank you. Glad to be on board.
Yeah, thank you, and a final congratulations. Is there anyone else on that would like to update us with their progress?
This is Tracy Bishop from California, and we started SMA last Wednesday, June 24th. So we're joining the club too.
Great.
Yeah. And is anyone from the lab on? We're using the qPCR CDC method. And, it's been 4 or 5 days, so I think we're about 6,000 or 7,000 samples. No positives yet.
And how many was that screened again? Sorry.
I forget what we're up to. Somewhere between 6,000 and 8,000.
Okay.
That we've so far. Yeah. But literally, it's only been 4 or 5 days.
This is Cindy from. Sorry, can you hear me?
Yeah.
So this is Cindy from the California lab side, and yes, so we are using the CDC qPCR method as well, and yep, by the end of this week we'll have around 10,000 screened.
Congratulations.
This is Bob. That's again just wonderful to hear on behalf of the newborns. And I'm curious methodologically, which instrument, qPCR, real-time PCR instrument, are you using, given the high throughput that's required there?
So this is Cindy again from GDL here at CDPH. And so we have 2 high throughput liquid handlers that can handle up to 4,000 samples, 3,000 to 4,000 samples maximum a day. And we are using a 384 well platform that will go into the Quant Studio 7 made by Thermo Fisher. So we process about up to 8 384 well plates per day.
That's just fabulous. I mean, that's a real accomplishment because you don't have a lot of material to work with in those small well microtiter plates. That's wonderful.
Yeah, it's 18 months of planning and lab modification and validation that went into this, and so far so good.
Wonderful.
Awesome. Well, thank you for that update. Is there anyone else on the line that would like to provide an update? All right, I'll take that as a no. Yeah, just congratulations to everyone for going live, and it sounds like everything's going well, and this is a great progress in the effort to get SMA countrywide. Okay, so is there? Oh, hello.
Sorry, I tried to unmute. This is Dr. Kupai from Lab Director at GDL. Thanks to Cindy and Tracy, they gave a quite good update. But I just saw the note that has been written. The 8 plates that has been said there is 8,384. Technically speaking, every day we have 24, at least 24, about 2,000 samples a day comes to California newborn screening. The robotic system that we have developed, it's not just a liquid handler, it's a full extraction process which takes the punch and just give at the end the 384 sealed with optical seal, which requires for Q7, which it's less hands-on process. And when we develop this, is as Cindy mentioned, thanks to Cindy and her group that she has spend a lot of time, particularly in this area, to validate. But this particular design, it's quite unique. It's only as far as when we started the process, we discussed it with Hamilton, and we said we wanted this. We had gave this specification, and we developed it together with Hamilton Robotic. It just goes through the whole process, and so far so good, no major issue. Yeah, that's otherwise we would need almost 3x times of staff and personnel to do the workload we have. But currently it's been quite fast and smooth. Okay.
That's awesome.
Yeah,
and I just wanted to confirm.
From other state that had this SMA, and I tried to unmute myself during the call. Unfortunately, I wasn't. We have gone live since June 24th. We haven't had any positive. We don't. I mean, if we go based on the published incident, we should have about 50 cases per year, which means around one a week. However, I do know that the incidents were quite low in New York at around 1 to 20,000. So I just wanted to know from experience of others. I do know that this is a random and might occur any time. However, just to get some idea of per 10,000 or 20,000 sample, any comments that what do we need to anticipate from other labs, from positivity rate and true positivity rate?
Hi, this is Mary Schroth with Cure SMA. So we've been collecting data from state labs and information from 6 labs that have been screening for more than a year. The incidence is 1 in 15,000 averaged among those labs.
1 to 15,000 positivity rate.
1.5, yes. But again, that's only 6 labs that have been screening for more than a year. So it's very preliminary. And the ranges that we have seen have been approximately, and I'll share this with you next month, but approximately 1 in 9,000 up to 1 in 30,000.
Okay.
So it's quite a wide range. And we can speculate about why that is. But we're learning yet, so as to what that incidence is across the country.
Sure. Thanks for the information. That's good information. So
and we will share this back with each of the state labs as we reach out to you and ask you for additional data. We're working on a reporting structure to give you information back. Thank you.
And Mary, can you remind me, sorry, I missed it. What was the sort of thinking about incidents before screening?
1 in 11,000.
Okay, thank you.
Mary, that's wonderful information. I have one thing to add, not specific to any disorder, but I began a few years ago wondering if the incidence or birth prevalence of a condition is N, 10,000, say, 1 in 10,000, and you screen 10,000, what are the chances that you'll get positive? So I couldn't figure this out myself. And there's a neat little trick to it, which I won't tell you today, but maybe someday. Anyhow, I went to someone who was much smarter, and it's fairly easy to do. So I did. So the answer is that if you have a probability of N in a population and you sample out N people, elements from that population, the chances of finding one, of getting a positive screen in our case, would be about 2 out of 3. It's 66 point something %. So, and it doesn't matter what the N is, if it's the same. I built a big spreadsheet and just did brute force calculation on it. So I don't know how to model that. Maybe I'll look into it. But I've never heard that information in any other setting about, if you're going to do a pilot screen, how many would you have to? So at screening the number that is the prevalence baseline, then that gets you about 2/3 of a chance of finding an affected baby, and you can do the calculation for any combination of numbers. So you could go to whatever probability you want. If you want 90%, it's got to be more babies.
Mm-hmm.
That's a Poisson.
No,
yeah. Thank you, Bob.
No, it's actually it's not statistics. It's raw probability.
Oh, okay.
And I'll tell you what the secret is. If the ability, how do you add probabilities? I know how to do an and probability, but I didn't know how to do an or probability. And the secret is an or probability is 1 minus the and probability. So you calculate the percentage of babies that you wouldn't get a positive test on and subtract that from 1.
Yeah, the other way is to do a Poisson probability where the population mean is actually the 1 over 1,000 or 1 over 10,000. It's pretty straightforward as well. But your way is good too. It's great.
Yeah, I mean, it's just a raw probability.
Yeah.
It doesn't make any assumptions about distribution.
Yeah. No, I get it. Thank you.
Awesome. Thank you. Thank you for explaining that. Okay. Is there anyone from APHL on the line that would like to update us on their progress? Okay, I'm not seeing anyone on the call right now, but luckily I reached out beforehand and was able to get a little bit of an update on the SMA XALD meeting. They were not able to reschedule the meeting due to the ongoing COVID pandemic, and so they will be transitioning some of their content into webinars. And so the first series is going to be about tiered testing with the dates listed there. There's no information out currently, but they will be disseminating that to the newborn screening community soon. And I guess if there's any questions, you can reach out to Kashia Hale at APHL. And with that, I guess we can conclude. Again, congratulations to everyone that has gone live in the last couple of weeks, and I hope that you all have a smooth process easing into it, and I hope you guys all have some great results for us next month on the call. Again, next month we will be hearing from Mary Schroth and Cure SMA about the ongoing push to bring SMA screening to nationwide levels. And so we will be here again the first Thursday of August, and you can find all the other recordings of these meetings, including last month's presentation by the Mayo Clinic team, at our YouTube channel. You can either follow this link or you can just go to YouTube and search for NBSTRN. We're also starting up a few other webinars about NBSTRN if you'd like to learn more about us. We have one posted currently. That was a presentation by Amy entitled, "What Can NBSTRN Do for You?" So give that a look. And yeah, so thank you all for attending, and look forward to next month's call.
Thank you.
Thank
Thank
you.
you.
Kevin.
Have a great holiday, everybody. Bye.
Thank you.
Yeah.
Thanks, you too.
Happy
Bye.
4th. Amy, I sent an email to you. Did it arrive?
I think she might have hopped off already, but I'll double check with her and ask her to reach out to you.
Okay, Kevin, I have your email somewhere. I will send that to you, and I'll also send it to Mary. I should have thought about that. So I'll get that out within the next few minutes.
Okay, that sounds perfect. Thank you.
Thank you all.
Thanks. Bye.